So I start with a few photos from me aged sort of 59 through to 62 and shows I've lost a little bit of weight. And we go into a sort of hypothesis as to what causes human or other animal bodies to not work so well as people get older. And the essential hypothesis here is that this really comes in the main from the failure of the genome to function. That is, the genome, the DNA or the ribosome, that sort of stuff, is something that produces proteins. And what we're saying is essentially that fails, and the cause is metabolic in nature. There are problems with damage to DNA, but I don't think that's the major part of it. And then obviously there's a question of what can you do about it? What then changes how you function? And there are tools to deal with that. So I'm going quickly because I haven't got that much time. And so the first question you have to ask with a cell is, you've got the DNA, which is used to produce mRNA, and then you've got the ribosome that uses the mRNA to produce proteins. And you need a mechanism to manage the usage of energy, so you don't just produce a load of mRNA transcripts and then there's no energy left to produce the protein. And transcription is done by an enzyme called RNA-Pol2, RNase polymerase 2. And to cut all of this short, right, the text is all here, but in essence, the DNA is opened up by adding acetyl-CoA to the histone. The histone is a protein to which the DNA is attached. But when the DNA is opened up, it enables transcription to occur. And if it's not opened up, then transcription does not occur or is slowed down. And there are a number of papers. In fact, if you look on my blog, johnhemming.blogspot.com, you can see links to this, or the citrate.science website as well. And there are a very number of papers that report that as people get older, as creatures get older, longer genes fail to be transcribed. That is, we don't produce the longer proteins. And there are alternative hypotheses for this. There are the ones about damage to the DNA, which I don't think is true, because it would just fail rather than be proportional. And then there's one relating to splicing factors, which that's from Lorna Harries, who's a professor at Exeter University in England. And then there's my approach, which is the substrate limit. Now, in fact, my view, which is the substrate limit, which is acetyl-CoA, is very similar to the splicing factors limit. Again, cutting things short, very often genes can produce a number of different proteins from the same gene, and that's called splicing, where different groups of bits of DNA are spliced together to produce a particular transcript. And there is good evidence that the level of cytosolic acetyl-CoA drives the splicing decisions. And when you actually look at some issues of aging, prostate cancer is a good one to look at. You can see prostate cancer is very strongly driven, or prostate BPH is strongly driven by aberrant splicing, that the splices that are produced are the wrong splices. They're not the splices you want to be produced. And there's a very good argument that, in a sense, that is a driver of development. So when you consider development, and if you consider aging as part of development, if we consider, for instance, when people go through puberty, precocious puberty and menopause, very often precocious puberty, menopause, and aging are related. So that if somebody, obviously a woman has precocious puberty and then has an early menopause, the men don't have the same sort of bright line distinction in development, which is a lot of development is not bright line. You can't say there is a specific point at which things change, but some development is bright line. And so the reason I don't think it's DNA damage is that would stop things. And as I said, the acetyl-CoA shortage is known to drive splicing decisions. Now, acetyl-CoA is a chemical, that's a molecule that exists in the mitochondria and outside the mitochondria. It's CoA means coenzyme A, and actually it indicates energy availability. And there are two main sources in the cytosol. One is citrate converted by the enzyme ACLY, citrate lyase, and the other one is from acetate, the enzyme ACCS2, and it can be used for a number of different things. But rather than go into all of that now, I would say mainly the main source of citrate is actually out of the mitochondria, and there is a protein that's kept in the inner mitochondrial membrane wall called the citrate carrier, also known as SLC25A1, and that passes citrate out. It in fact swaps citrate for malate plus a proton. That's, you add a proton to citrate and swap it with malate. And the important thing about the level of acetyl-CoA is it indicates the power available in the cell. So if there's more acetyl-CoA, the cell has more power, it has a greater ability to produce proteins, as well as do lots of other things, because there are obviously other things that are done by acetyl-CoA. So interestingly, a higher level of citrate in the serum, in the blood, is also pro-longevity. And you can see when zygotes are fertilized, citrate is used to increase acetylation and facilitate transcription. That's the primary function, in fact, of the prostate gland, which is to put citrate in seminal fluid so that when the egg is fertilized, it fertilizes and a rapid rate of protein production can occur. And there's been experiments done with the Drosophila melanogaster, with the MINDI gene, which is a version of SLC13A5, also known as I'm not dead yet. And that shows that there is a merit in additional dietary citrate. And there is a direct experiment done, not in human beings, in fruit flies, which saw that additional citrate in the diet. And we all eat citrate. We probably eat about 4 grams of citrate a day on average. So it's not as if we don't eat it already, but additional citrate is pro-longevity. And the one part of the hypothesis is that the reason there is a shortage of SLC25A1 in the mitochondrial membrane wall is because there is part of SASP going around. Now, I think people know about senescent cells. Senescent cells are sometimes described as zombie cells, but senescent cells are cells in the body which are not functioning correctly. And you often see it, like in the pancreas. If you get a load of senescent cells, they sit there absorbing fat. And if you get too much fat, your pancreas doesn't work, and that's obviously not good news. So part of the hypothesis is a cause of senescent cells is that there is a shortage, in fact, of SLC25A1 in the mitochondrial membrane wall, and that then causes there to be insufficient acetyl-CoA for the cell to be correctly differentiated at the early stage of differentiation, where stem cells get converted into somatic cells. And there's a good paper which is linked to here, which looks at how osteoporosis occurs from a shortage of cytosolic acetyl-CoA. It's still available in the mitochondria, but it can't get out of the mitochondria citrate because of a shortage of SLC25A1. Now, osteoporosis is well known to be caused by the failure to differentiate the osteoblasts. There are 2 types of cells used for maintaining bones: osteoblasts and osteoclasts. Osteoclasts reduce, they tear down the bones, and osteoblasts rebuild the bones. And normally, over a period of 7 years, bones are renewed. But as people get older, because of the shortage of osteoblasts, which is known from this paper to be caused by a shortage of acetyl-CoA in the cytosol, which is caused by a shortage of the citrate carrier in the mitochondrial membrane wall, that causes osteoporosis because the bones are chewed up and not rebuilt. So that's that. And there is actually a very good paper which was produced by a Portuguese student, which looked at the fact that interleukin-10, which does reduce the levels of SLC25A1, does cause senescence. So if you look around the body, in the kidneys, excessive senescent cells causes chronic kidney disease. In terms of maintaining bone structure, senescence replacing cytoblasts means not cytoblasts, osteoblasts. Senescence in that situation causes bones to deteriorate and not be rebuilt. And this is a good paper, which was probably unprintable in the normal journals, because the journals would say interleukin-10 is anti-inflammatory, which it is, and therefore it can't be interleukin-10. But if the evidence points to it being interleukin-10 that causes senescence, we should understand what the mechanism for that is, rather than just say, let's not tell anybody. But it's there, and again, I'll give you the link to the paper there. So there is potentially also an issue with translation, although I think most of the issue lies in transcription, and that relates to a failure of translation from a shortage of ATP. All right, so we have this question, therefore, as to what the mechanism is that drives all of this. And there has to be a system to prevent longer genes functioning, because otherwise what you would get is ATP exhaustion, and you wouldn't actually have the energy to produce the proteins. And we can look at creatures where the transcription process is modified, and the very good ones, EU social, that is hive creatures, Hymenoptera being bees and ants. All right, so a lot of people will know about the queen bee, and the queen bee is obviously when you've got a hive of bees, the queen bee produces all the eggs. And the reason why the queen bee is different to the worker bee isn't the genes. The genes are exactly the same. The queen bee eats royal jelly, which is produced by the bees every day of her life, whereas worker bees only have it for 3 days. And the difference with that is that epigenetically, therefore, different proteins are produced. And their different proteins are produced partly through increased availability of coenzyme A, but also partly through the inhibition of the HDAC. Histone deacetylase inhibitors. You have an enzyme which is called a histone deacetylase, and that removes the acetyl group from the histone. So it closes down the DNA, so the DNA then cannot be expressed. And if you delay that process, then more protein is produced. So, and it's quite interesting, you see the difference in lifespan between a queen bee that lives, say, 2 to 3 years, and a worker bee, which is in the honey season, doesn't live very long, about 6 weeks. And obviously the circumstances are different, but the queen bee is going through a lot of work laying all the eggs. So interventions you can do on transcription are to increase cytosolic acetyl-CoA, which can be done through acetate and citrate. Obviously, acetate is, you've got citric acid, which is lemon juice. I'm not a fan of eating acids. And acetic acid, which is vinegar. Those, when they're with a cation rather than with a proton, are in the form of a citrate or acetate. Interestingly, the Janus kinase inhibitors also have that effect. The Janus kinase is the interface in the cell wall that links interleukin-10 and various other interleukins to internal cell focuses. And then you can slow down deacetylation through using HDAC inhibitors. Another area which is perhaps even a better target at times is also to improve the mitochondria. Now, again, mitochondria, we've got these things in our cells which theoretically many billions of years ago were bacteria coming into our cells, and they generate energy, and they're very good because they create much more efficient energy production. Very interestingly, the mitochondria have their own DNA. So some of the proteins in the mitochondria come from nuclear DNA, but some of the proteins in the mitochondria, particularly those for generating energy in the electron transport chain, are the DNA for the mitochondria is actually in the mitochondria. And it's unusual in 2 different ways. One is it doesn't have a histone, so it's very vulnerable to damage. And the other one is it's a circle because it's bacterial DNA historically. And also you have multiple copies in an individual DNA. Now, an interesting thing about it is not only can you have different mitochondrial DNA in different mitochondria in the same cell, but actually you can have different mitochondrial DNA in an individual mitochondrion. And as the mitochondrial DNA gets damaged through reactive oxygen, for free radicals, if people want to call it, that can make the mitochondria less efficient. And you get what's called heteroplasmy, meaning variations in mitochondrial DNA. Now, we have a lot of things, rapamycin, urolithin A, exercise, fasting, and PEMF, the long list of them. You all know these things. And what these do is they encourage the cell to recycle damaged mitochondria. And what the cell actually does is it picks on the mitochondria which have the lower mitochondrial membrane potential. And the mitochondrial membrane potential, which is a voltage across the mitochondrial membrane, links to the efficiency of the mitochondria. That is the rate at which it will convert ATP from oxygen. And so as you encourage the recycling of mitochondria in whatever technique that you use, you end up with the cell getting rid of the mitochondria that are less efficient. And then the cell says, I've not got enough mitochondria. And it starts creating new mitochondria, but it creates them by creating them with copies from the current mitochondria, which are more efficient mitochondria. So this is essentially why all the techniques people use to improve the function of the body, like exercise, like AMPK activators, etc., are techniques that improve the mitochondria and the average efficiency. Now, I've been doing a small human trial, mainly myself, on using these things in a synergistic manner, because one of the difficulties with all the interventions is that you can have lots of different interventions, but you can't be quite sure which are synergistic. And if you look at Aubrey de Grey's current work with the robust mouse rejuvenation trials, they're doing some good work on looking at what works on a combinatory basis. But if you don't have an idea as to the mechanism, it's very hard to work out what is synergistic and what is not. But I've got some useful results from my own work. For instance, C-reactive protein. Now, C-reactive protein is produced from interleukin-6, and interleukin-6 is part of SASP. So if you can get like your C-reactive protein down below the measurable limit of 0.15 milligrams per liter, which is 0.015 milligrams per deciliter, that's quite a good result, all right? And then if we look at the changes in terms of glucose handling, that's also quite a good result. And cystatin C here, again, a value of 0.76 is a good result. I'm not a massive fan of the epigenetic age things, but if something's going to say that I'm 36.58 years old, I'm going to buy at that because I'm 64. Well, here is 63, 66. But these things vary, different things. An interesting thing about additional citrate supplementation is it encourages cellular repair, and it will gradually regrow hair very, very slowly, and it helps with building muscle mass. So, and here are a few of the links to the papers. Now, I've gone through that really very quickly, simply because I'm going to have to finish soon, and I thought if I do it too slowly, you wouldn't have time for questions, and often questions are the better time of discussion. So I shall end that now, and we can go back to where we were. Yeah? Have I ended that? No, I haven't ended it, have I? I've got to stop presenting. There we go. That's gone now.
Nice. Thank you. Thank you, John. Lisa, do you have a question?
Yeah, John, what is the role of glutathione?
Well, glutathione is another antioxidant. There are lots of antioxidants. Now, actually, personally, I think the best antioxidant is melatonin, because in the mitochondria, if you want me to talk a little bit about melatonin, in the mitochondria, melatonin is generated from serotonin in the mitochondria. So one of the things, for instance, that infrared light does is it encourages the production of serotonin, and that can then be converted into melatonin. And melatonin as an antioxidant can reduce something like 9 or 10 species of ROS, where a lot of the other antioxidants can only reduce 1. And a very interesting thing is that children have a very high level of melatonin. And I think one of the reasons, what actually happens is the body is producing melatonin in all of the cells, as well as in the pineal gland. And overnight, there is melatonin placed in serum by the pineal gland that goes around the body. And I think one of the reasons why melatonin is thought to hold back puberty is it holds back the damage to mitochondrial DNA. And there's no question about it that it's a very good antioxidant. So glutathione, yeah, there are loads of antioxidants. There are interesting questions as to which antioxidants are actually ones which enable people to have greater. I've got a cat here. You can see the cat. All right. But there are interesting questions as to which antioxidants actually are pro longevity, because some antioxidants are seen to be not pro longevity. But so glutathione, it's a useful antioxidant, but I wouldn't say it's necessarily going to make a massive difference.
Okay. And I'm sorry, I missed, what is your website or?
Well, I've got various ones. I'll put some links through. Let me just get, I've got Citrate Science, which is probably, let me get a link up to a paper I put up on there. I presented it at the British Society for Research on Aging as a poster there. So let me just get that into the chat for you. So this is one of them.
Thank you.
All right. That's probably good because it gives you links to all the other papers that are referenced, and it's, you know, going through this, if you're not used to all of this, it's quite a lot of new information, and it's worth reading all the background papers.
Beautiful, thank you.
So would you advise for taking melatonin supplements in the case of someone struggling with sleep?
Well, melatonin is complicated because it has this interplay between melatonin and the autonomous nervous system. And what actually, it's not so much a gamma aminobutyric acid. Is you've got within the brain, you have the sort of glutamate GABA balance. The cat's jumping around again. Wants to feed it again. She's had her lunch. She's not going to get another lunch. A lot of things people take for the purposes of sleep are things which are GABAergic. That is, they tend to sedate the brain. Melatonin is very mildly GABAergic. Its main effect is its impact on the autonomous nervous system, which can be either, this is the business about HRV. If you're more switched to being parasympathetic state, your HRV will be higher, and you'll be more, your cortisol levels will be lower. If you're in the sympathetic state, then your cortisol levels will be higher. Now, melatonin actually inhibits cortisol, ACTH, and CRH, which form the HPA cycle. And everyone knows they've got circadian cycles, which are the daily sleep cycle. People tend not to be so aware of the HPA cycle or the sleep cycle or the autonomous nervous system cycle, which operates roughly every 90 minutes. Now, with melatonin, if you take melatonin close to the start of a sleep cycle or close to the start of an HPA cycle, then it will likely ensure that you're in the parasympathetic state with a low level of cortisol, and therefore able to sleep. If you take melatonin, say 20 minutes into the start of a cycle, because its half-life in serum is about 30 minutes, it will be dying off by the end of the sleep cycle, and you're likely to go into the cortisol awakening response state with the autonomous nervous system in the sympathetic state, and that will wake you up. So melatonin can put you to sleep, and it can wake you up. And what you have to be, if you're wearing a sleep tracker, you can often see where the sleep cycles are. Melatonin can be particularly good if you wake up in the middle of the night, but you shouldn't take it straight away. You should wait until you ideally see where your sleep cycles are, because very often if your tracker will tell you where you move into REM and various things like that. Ideally, what you want to do is wait a little bit until you're close to the end of a sleep cycle, but it's not going to be exactly the same for everybody. And then at that point, it will make sure you get into the parasympathetic state in the next sleep cycle, rather than being in the sympathetic state with cortisol coursing through your veins and keeping you awake. So it is good, but it's complicated. I take a lot of melatonin. I take around about 1.5 grams a night. The reason I take so much melatonin, even if I'm not having problems sleeping, is because I want to top up the melatonin in my mitochondria and reduce mitochondrial heteroplasmy. I don't take it primarily for sleep, although sleep is a good thing.
Interesting.
Sorry,
And
that's quite a long answer, but
thank you. What would be the best sources for additional citric supplementation?
well, I mix sodium citrate. It may sound odd I supplement sodium, but I do. My blood pressure is quite good. My blood pressure is like, good blood pressure. It's got a slightly high pulse pressure, but the average arterial pressure is below the 120/80 that people see as being acceptable. Remembering I'm 64, and I'm not a massive gym bunny either. So the thing is that you've got sodium citrate, magnesium citrate, potassium citrate, and calcium citrate. I mix them together in a ratio, because if you've got to get the balance between potassium and sodium right. And I aim to have about 10 grams of citrate a day supplementarily. In theory, people supplement. They eat about 4 grams a day anyway. So but it does shift that sort of thing. But you've got to be very careful, because it does put pressure on your kidneys. So you have to monitor the renal markers and make sure you're not causing yourself a problem. So it's a complicated thing. Again, citrate has a half-life of about 30 minutes. So it's another one of those things where you've got to time it right. It's not the same problem as you have with melatonin, because you haven't got this sort of HPA cycle issue going on. But you need to, what it does is it'll top up the acetyl-CoA for a period of time, and then it'll die down again. Because you have citrate in your blood anyway. It runs at around about 100 micromolar most of the time. So you take it up a little bit, then it pops back down again.
And are there natural sources of food that can help with it?
Well, there are, because there's lots of things with citric acid in them, but most food will have some citrate in it. But the danger is you don't want to be too acidic. I think there's an interesting paper recently which pointed to the serum acid indicating aging. So as a biological measure, your body deteriorates the more you're acidic, right? So you really, citric acid, lemon juice. So you've got lemon juice in lemons, limes, citrus fruits, obviously, grapefruit. The problem with that is you're having the acid as well. So I would be careful about that. But it's easy enough to get the sodium citrate. They're all foodstuffs. They're in the UK, they have E numbers because they're foodstuffs.
Do you measure these things as well, or what do you measure?
Well, I measure a lot because I do a blood test every week. So I'm measuring all of these different things that, the standard blood test with, the standard range, full blood count, white, in terms of white blood cells, all those sorts of things, kidney markers, liver markers, testosterone, CRP, etc. So very much, if you don't measure things, you don't know what's happening.
Yep.
But I also monitor sort of hair regrowth, because I was completely bald on the top. Now that was already there, but this wasn't. All this stuff here. And that it's a great getting the mitochondria fixed in your if you've got male pattern baldness, as I do, what that means is your mitochondria and your scalp around here get very badly damaged over a number of years, and repairing them is very hard. Keeping them going is a bit easier, but that's hard, because you've got to get the mitochondria fixed. It won't just work. You can't just eat a bit of citrate and all your hair suddenly grows back and you look like you're a teenager again. It doesn't work that way, as you can tell from me. But you can see where I started out. So, in many ways, the function is quite good. So if your body is more flexible, if you don't have the aches and pains, that's quite good. That's a useful situation. but it's complex because you're working on the whole body as a system. So you're working on how you can improve all your mitochondria. This is where rapamycin comes in as well. Rapamycin is an mTOR inhibitor. It encourages mitophagy, so you chew up the inefficient mitochondria and you replace them with more efficient mitochondria. But obviously, you're all quite a lot chronologically younger than me, so you shouldn't be taking rapamycin all the time, because it's an mTOR inhibitor cuts out the anabolic phase of the body as well. You've got to go in through a catabolic anabolic phase, and the catabolic phase for somebody in their 30s or 20s is perhaps once a year. You don't want to do a spring clean every week.
No, that's a statement. That's by all rapamycin. Do you know of any other mTOR inhibitors that, because obviously rapamycin price or it's yet understudied, the dosages to get that right. There are some drawbacks or challenges with that. Do you know of other methods for inhibiting?
Well, the thing about rapamycin is there's a lot of evidence on rapamycin, so you know where the safety levels lie. Now, there is a forum online called Rapamycin News, which you can see various things in there. And I've done experiments where I've taken a proper large dose of rapamycin, whilst wearing a CGM and doing white blood tests twice a week. So you can see all the movements in white blood cell counts and average glucose levels, because the effects of rapamycin are very, very well known. Now, it has a half-life of 60 hours. It's a bit long, really. If you want to have a catabolic cycle, you want it to be a short cycle, and then move to the anabolic phase separately. So it's a bit long for that, but I'm happier with a drug that we have a lot of evidence. We know where the safety, we know what the side effects are, and there are quite a wide range of side effects. But they are quite well known. So, from a safety point of view, it's reasonably straightforward. But we are aware that it affects lipids. We are aware that it affects glucose. We are aware that it affects the white blood cell count. So you can have issues with infection when you take rapamycin. Now, we know all of that, but we also know it does drive greater autophagy. That's a good thing.
You mentioned it's not a good idea to make the body acidic. And how does that work? Because once you consume something, the stomach pH will adjust to what you consume. And if you put a lot of acid, it's going to get a bit basic. And if you put a lot of basic, it's going to get more acidic just to compensate for what you put in. So how do you propose go about this?
Well, there are different foodstuffs that cause different effects, and in the end, your body adjusts. It aims to keep the pH of cells to around about 7.5, which is obviously very mildly basic. One of the challenges, if a cell finds itself getting a bit too acidic, what it tends to do is to deacetylate the histone. Now, that's something we don't want to do because we're reducing protein production. I personally think a useful thing is to measure urinary pH, because it shows you what your body's trying to do. And it may sound a bit of an odd thing, but at times my urinary pH ends up about 9, which is really quite high. And there was a very good paper recently that looked at mortality linked to acidity. Now, yes, you're right. There's different, even in a cell, different parts of the cell are going to be at different pHs. And if we look at the mitochondria with the proton pump in the mitochondria, protons, hydrogen ions, are a key part of energy production. But I think there is good evidence. You really don't want to be feeding your body with so many hydrogen ions, protons, that are as ions, such that it has to handle getting rid of them. And look at kidney stones. If you take citrate, which makes you alkaline, it reduces kidney stones, for instance. So there's a lot of evidence that you can be much more healthy. And people go into drinking alkaline water. you know, if it's very mildly alkaline, probably not such a bad thing. But I'd prefer to stick with things like citrate or acetate. Citrate definitely, and that as I said, that I know because I measure my pH of my urine from time to time, and it has been as high as 9, which is very high. But obviously, different things do different things. And, you know, alcohol. I do drink alcohol, and that can make you quite acidic. But alcohol, the thing about the metabolism of alcohol is you've got this first step of alcohol, ethanol going to acetaldehyde, and the second state of acetaldehyde going to acetate, and it does switch urine to a sort of like pH of about 6, maybe 5 sometimes. So you've got to watch these things.
Have you experimented or dealt with caffeine at all? So I have quite a complicated relationship with caffeine.
Yeah, caffeine. I drink tea. I don't tend to drink coffee. Caffeine is one of a very large number of substances, like methylene blue, for instance, which I have used as well, which are argued with some evidence to increase mitochondrial efficiency. And I think when you look at the caffeine, what's its half-life? About 4 hours, about 4 to 6 hours, something like that. So it's a bit of a challenge with sleep, because obviously if your brain's got a lot more energy in the middle of the night, you don't sleep so well. And there's a lot of, like another one, menaquinone 7, also vitamin K2, right? Vitamin K2 has a number of different forms depending on how many isoprene residues there are. And menaquinone 7, which unsurprisingly has 7 isoprene residues, is also something that makes mitochondria more efficient. So that will keep you awake as well. There's a very interesting thing about certain substances which make mitochondria more efficient that you can see that they do because you can't sleep. If it gets in the brain. But caffeine, yeah, is caffeine pro longevity? It probably is. Do people badly react to it at times? Some people do. Does everyone react the same to everything? No. Different people react in different ways. Should you avoid caffeine late at night? Probably yes. It doesn't really help with sleep. Is it a magic substance? Probably not. It's probably quite helpful. I would think methylene blue, maybe. Again, methylene blue, low half-life. Again, probably lower than I don't know what methylene blue is actually, but it's an interest. I have played a bit with methylene blue, not really got much from it. I think menaquinone 7, that has quite dramatic effects. That's vitamin K2. And that has quite, but that's menaquinone 7, not menaquinone 4. When you get supplements, you can get menaquinone 4, menaquinone 7, and menaquinone 9 in the supplement form. In cheese, you can get all the way up to 13 different types of cheese, different amounts. Maybe why cheese sometimes keeps people awake at night, because of the menaquinones in it. But there you see it's adding an extra electron carrier for the electron transport chain, so it's the electron acceptor. So that's how that works. Similar to methylene blue, really.
John,
Do you want to experiment with these things?
I had a question about like autophagy, like in terms of the autophagy that's triggered by rapamycin, and let's say if you do like a 3 to 5 days fast, is there any kind of like data inferences have you done? Like, is it the same amount for somebody who's like, let's say in late 20s?
That would be good to know. So one thing I want to experiment with urolithin A to see to what I've got some of it. I've had a bit of a play with it. Rapamycin has very, very, very obvious effects, and as I said, I've taken rapamycin whilst wearing a CGM, whilst doing blood tests twice a week, and that's published on Rapamycin News. Shall I try and find that whilst we're sort of able to do this, because I'll put it in the chat. It's quite a useful thing because I don't think that information is available anywhere else. I can't say what I'm actually sort of in a bit of a fast at the moment. As I'm drinking cups of tea with milk, which is not entirely a fast, but let me just see if I can get it up. Yeah, here we go. This links to let me get that up again. Yeah, here we go. That's in the chat. That gives you all the stuff on there. I don't know how it compares to fasting. Fasting, I think one of the points about fasting is generally you've got to probably hit 3 or 4 days before you start really having major autophagy effects. Although there is an argument that even intermittent fasting overnight has some autophagy effects as well.
So if let's say I want to experiment like single dose of rapamycin and let's say like 3 days or a 5 days fast, what are some of the blood work that I should do before and after?
Well, I would always get a basic CRP to give you an idea what's going on there. The problem is that what would be really useful is the measures on the acidity of the blood serum. I don't think that's easy to get, sadly. Well, you can see what I've done in the it's a standard, because one of the issues is obviously cost. So if you're doing like weekly blood tests or blood tests twice a week, you're going to go for the standard blood test rather than additional ones. I may get a rapamycin serum test at some stage, but in many ways I'm more interested in the link between the dose of rapamycin and the outcome. And obviously you're talking about cytochrome P450 inhibitors as well. So there are enzymes in your intestines that metabolize rapamycin, and if you take a grapefruit or a pomegranate or a pomelo, you can inhibit those enzymes, so it becomes more bioavailable. That's quite a useful process. Also reduces the cost of rapamycin if you're taking quite a high dose of rapamycin. But that depends on the amounts, because my dose. They did an experiment called the Pearl Try, where people were taking the effective dose of 3 milligrams of rapamycin. And my single high dose, when you take into account the multiplier effect from the grapefruit, was about 77 milligrams, which is a lot more. But there's only one off, you see, so it wasn't. I don't take it. I take it normally about every 6 weeks, because most of the time I want mTOR to function normally.
Have you participated in the Pearl trial?
No, I wasn't in the Pearl trial. No. Look, it was a nice idea, but what the problem is, what did it prove? It proved if you don't take much rapamycin, not a lot happens. We already knew that. One of the difficulties, it was damaged. They meant well, but they were damaged by the need to have a masking process. And but because they wanted a placebo, they went to a compounding pharmacist. And when it was compounded, it lost the enteric coating, and therefore the bioavailability was exceedingly low. And all they found out was that rapamycin is safe in small doses. And we know that. The safest daily dose is deemed to be about 40 milligrams. I think that's with enteric coating. I don't know for certain, but I would assume it's with enteric coating, because most rapamycin has enteric coating. That's daily, though, you see. That's equivalent to a single dose of about 160 milligrams in terms of peak serum concentration.
What about the urolithin A you mentioned? That sounded like a little experiment we could do.
Yeah, well, the thing about urolithin A, again, you see, rapamycin is a prescription drug. Now, in the UK, you can import prescription drugs for your own purposes. And there's no, as long as they're not controlled substances, there's no difficulty with that. In the USA, it's a little bit complicated, because in theory you're not allowed to, but in fact the FDA don't mind that much, but it might get blocked by customs. In some countries like Sweden, you can't import the thing at all, really. And so you get these complexities as to how to get things. Now, urolithin A is a foodstuff, so you can just buy it off Amazon. And so but the question then is, how much of an mTOR inhibitor is it? And I do have as one of my plans to put a CGM on and take some large doses of urolithin A and see if it pushes up glucose. Because if it pushes up, basically, when mTOR is inhibited in the liver, you get hepatic insulin resistance. And with the it's quite interesting. If you see my results that I've linked to, you'll see how I take rapamycin, I get hepatic insulin resistance. My blood glucose average goes up quite a long way. The insulin then adjusts and goes up. And then the rapamycin fades, the mTOR inhibition disappears, and the glucose goes right down. So the average glucose goes down to about 4.5. That's millimolar per liter, which is an HbA1c of about 4.6, 4.7. All right, which is pretty good from an HbA1c point of view if you're into that sort of, that's percent, of course. And then what happens is actually then it adjusts and comes back to normal. So after about a 6 or 7 week cycle, I'm back to where I was before I took the rapamycin. But actually, what happens in fact is my glucose handles moves into pre-diabetic and maybe slightly diabetic, and then it moves out of pre-diabetic into very much not diabetic at all, and then back to sort of my normal HbA1c is about 4.9, which is good for somebody of my age, but it's sort of it's not even pre-diabetic. It's well away from that.
That's good.
I need to finish in 2 minutes, so if there's anything else, there's a burning question, I'll try and answer it quickly. Otherwise, I'm going to have to disappear off the call.
Definitely, thank you from my side. It was really.
It's a bit of a sort of a rush through the whole thing, but it's interesting, and there's more papers coming out in this sort of area now. There was one that came out this week. I haven't managed to get a copy of it yet because I haven't wanted to pay the $50 or so which they wanted me to pay to get it, and I use Deep Dive, which is quite good for giving you a relatively, still quite expensive, about $500 a year, just to give you access to some journals, but it's not the same as paying a subscription to all of them.
What I'm learning here really is it doesn't less matter like what you really take, like the specifics of the each of the things you take, but more the systematic view on top of what is the context and how does
mechanistic. Basically, if you can get synergy, so you're not just randomly taking substance in the hope they'll do you some good, but you're synergistically aiming to improve your transcription, which is where all this Yankee TCN stuff comes in. But I've got to stop now. So I'm happy to talk. Obviously, I don't mind talking about this. It's just I've got to go and get my son from school and take him climbing. So
have a great day. Thank you so much.
Bye-bye,
Yes,
everybody.
thank you. Bye.
Thank you, John.